Good morning, good afternoon, good evening, whatever the time may be. You sat down to listen and to watch this pre-recorded lecture for your general zoology class. Now, I talked already about the flipped classroom concept at the beginning of the semester. Now, we will have our first pre-recorded lecture. Now, this is a class. This is your lecture, so please sit down and pay attention. Watch this class and take notes. Don't watch this lecture while you're drying your hair or you're cooking or you're clipping your fingernails. No, this is a class, so pay attention and take notes. Just to drive the point home a little bit, at the beginning of the class on Tuesday, we will have a short quiz. Now, the quiz will test you and it will tell me if you actually watched the lecture. So, the level of the quiz, well, it's just deep enough to tell me that you actually listened and you paid attention. After the quiz on Tuesday, we will have a question and answer session. So if anything is not clear after watching this video, you just let me know on Tuesday and we will talk about it. And once we're done with the question and answer session, we will get to the main part of the class, which is solving application problems. Okay, so today's class is about animal development. So in this class we are concerned with what happens from fertilization of a zygote, a single cell, that then starts to divide and differentiate and become more complex and produce a multicellular animal. Most of the material that I'm talking about you find in chapter 19. Now, chapter 19 has a section or has subsections on animals and subsections on plants. So whenever you read plant, just skip it. I want you to read the animal part. Okay, so what is development? Well, development is a process of maturation of an organism, and it involves growth, cell differentiation, that eventually leads to morphogenesis. Morphogenesis, well, that's the process of creating structure. We will talk about these different subsections in more detail for the next class. Really, today what we're concerned with is how do cells differentiate? How do cells become something that looks very, very different from what the zygote was? So today's key question is, how can we get a multicellular organism consisting of cells that are very different, specialized for different purposes? Now, this may seem a trite question at first, but consider this. All cells have exactly the same genome. So if they have exactly the same genetic material, how do they become different? And they all arise from one single cell, our zygote. Now, the zygote is the fertilized egg, and it will start to divide. We're going to talk about this in much more detail next lecture. It divides to become a little ball of cells, and eventually it becomes a hollow ball of cells called the blastula. But then things change. After the blastula stage, we get an embryonic form that's called the gastrula. And the gastrula is the first time in the development of an organism, well, let's be specific, in the development of an animal, that we're gonna start see differentiation of cells. It is at this point that the fate of different cells is already set in motion. After the gastrula stage, we see the formation of organs and organ systems and different tissues that eventually will make an adult animal. All right, so how does this happen? The zygote is your ultimate stem cell. It is the cell that can become just about anything. Now, the technical term that we have for that is, it is a totipotent cell. Now, even at later stages, depending on which animal we're talking about, we may be in the 8-cell stage, in the 16-stage cell. In many animals, the cells are still totipotent. That is, they can become just about anything. Now, you may have heard that even in adult human beings, we still have some stem cells, as in our bone marrow, for example. Cells that can differentiate and become blood cells. Now, those stem cells can become different types of blood cells, but they cannot just become anything in the body. They cannot become a nerve cell just like that. They cannot become a liver cell. They can become different types of blood cells. Now, in this case, the fate of these cells has already been partially determined. And in this case, we talk about pluripotent cells. Okay, that's just a little bit of terminology that we need to be aware of. Now, since the early days of genomics, the early days of genetics, we have come to learn that pretty much all the cells in our body have the same genome. Now, there are a few exceptions to that we're going to encounter later in this course, but for all intents and purposes right now, all our cells have exactly the same genome. However, they have a different proteome. That is, their content, the protein content, is very different. Now, in terms of development, we call this genomic equivalence. That means all of the cells have the same genome. They have exactly the same genetic material. Now, another thing that was discovered early on in the study of animal development was that it is often the cytoplasm surrounding the nucleus that plays in some way a crucial role in determining the fate of a cell. Okay, I want to tell you a little bit about an experiment that has been done for some time ago. In this particular experiment, and let me just pull out here the pointer, there we go. We start with a frog embryo. This is probably a late blastula, just beginning gastrula stage. At this stage, we already have identified different regions, different cell regions, that will become, if we let the organism develop, if we let the animal develop, they will become a certain tissue. For example, if we extract cells from this particular area here, or not extract, if we follow these cells and see how they divide into the future, we will see that eventually they become skin tissue. If we take cells from this area here and we follow them through all their divisions until we get an adult animal, these cells become brain tissue. And this area here in red, well, those are cells that, as the animal develops, will become the notochord. Now, the notochord is a structure typical of one phylum of animals, the Chordata. Okay, remember that. Notochord, Chordata. In lab, we will talk in much more detail about the chordates. that, well, ultimately we belong to as well. Now, the question was, I guess, at the onset of this particular study, well, how early in an embryo is the fate of the cell determined? So what the researchers did was, and this is pretty cool, they extracted cells from the early embryo. And in this case, cells that we know at this particular position, they will become skin cells. And we transplant these cells. We insert them either over here or over here. Those are the regions that correspond to these cells here, the ones that will ultimately become brain tissue and the ones that become the notochord. Now, if these cells already have a fixed fate, if these cells have already differentiated and their fate is sealed, then we would expect, no matter where we transplant them to, they will become skin cells. But what do we actually see in this experiment? These transplanted cells that we know in this embryo will become skin cells. We follow them through all the divisions in the growth and development of the animal, and lo and behold, they become brain tissue. And the transplant that we put right here that we insert right here in an area that we know is going to become a notochord, well, these cells will become notochord tissue. Now, what does this tell us? This tells us that the cell fates in this embryo have not been determined yet. So, these are pretty much totipotent stem cells. They can become anything else. Now, another lesson from this experiment is that obviously the location of where we transplant these cells to is important. So it's really important. Position, the exact location will determine what will become of these cells in an adult. Now the question is, well, how do these become different cells? And here we have one example that tells us at least part of the story. Here we have a myoblast. A myoblast is a pluripotent cell that will become a muscle, so it's a muscle precursor cell. And we know that the expression of a particular gene in this myoblast will actually convert this myoblast to an adult skeletal muscle cell. The gene that is being described is the MyoD1, and that stands for Myoblast Determination Gene 1. This particular gene encodes a protein that will trigger the transcription of other genes. Now, you may remember from Biology 2 that there are some very important proteins out there, or in our bodies, there are some very important proteins that actually help the DNA and RNA polymerases, well, in this case the RNA polymerase, to find exactly the spot from where transcription takes place. And not only that, these particular proteins will cause the transcription of a particular gene. Now, those proteins are called transcription factors. And we can already see that throughout the entire development of an animal, transcription factors are going to play a crucial role. They're going to play a crucial role, not role. Okay? Okay. But obviously, in this case, we can see, yeah, sure, MyoD1 will trigger the myoblast becoming a skeletal muscle cell. But what causes the production of MyoD1? Well, this is only part of the story, obviously. Now, it turns out that, as we've already seen with the experiment, the location of where the cells are is important. We could also say, in other words, that the immediate environment of where the cell is important. Now, a cell is going to have an internal environment in the cytoplasm, and there are a lot of things, very important processes that take place in the cytoplasm. But, well, and those processes will help in the process of differentiation, but there's also an external environment. An external environment, the extracellular matrix, or the interstitial fluids, all this fluid-filled space between the cells is going to be important. And it's easy to see how. There are obviously signaling molecules, and this includes, amongst others, hormones. We'll talk about those later. There are cell adhesion molecules. We'll talk a little bit about those for next lecture. Cell adhesion molecules that are also important in the process of differentiation. And finally, the ones that we are going to talk about today, there's a group of molecules called the morphogens. Now, the term already tells us in a way what these morphogens are doing, right? Morpho, that's form. Gen, creating. They are molecules that, in a way, create form. Okay, so let's turn now to these different signals. Now, first, let's look at an internal signal in what we call cytoplasmic segregation. At this point, what we need to realize, and this is very, very important, at this point, what we need to realize is that the inside of a cell is not homogeneous. It's not a little balloon filled with water or some sort of solution. We mix it all up and everything on the inside is the same. It's not the case. Actually, there are different regions within the cytoplasm. They are regions that give us what we refer to as cell polarity. There's a pole up here, it's the animal pole. There's the nucleus. The nucleus is always close to the animal pole. And then we have the other pole, which is the vegetal pole. Okay, be careful, it's the vegetal, not the vegetable pole. Okay, vegetal pole. So on the animal pole we have the nucleus, and the vegetal pole we have nutrients, different types of nutrient stores. Now, not only are the nucleus and the nutrient stores unevenly distributed in the cytoplasm, but we also have certain molecules that we call cytoplasmic determinants. Cytoplasmic determinants, which may either be mRNA or proteins that are unequally distributed within the cytoplasm. That means that if a cytoplasmic determinant tends to be up here near the animal pole, after some divisions, we will get some cells that contain the cytoplasmic determinant and others that don't. Or the cytoplasmic determinant may be in the vegetal pole, and in this case, after subsequent divisions, some cells are going to have the cytoplasmic determinant and others don't. Now, the cytoplasmic determinants are obviously going to be important in triggering the transcription of certain genes. So if we have a cytoplasmic determinant down here in these two cells, but not these, already different genes may be triggered in these cells from these ones up here. And these may have some cytoplasmic determinants not shared with these cells. So already by unequally distributing these cytoplasmic determinants in the cell, we are setting the stage after subsequent divisions for differential gene expression. Now, these cytoplasmic determinants, which again may be mRNA or protein, they are the product of maternal effects genes. That is, these genes are produced, or are expressed rather, in the egg cell that is produced by the mother. And only by the egg cell. So these particular determinants, cytoplasmic determinants, come from our mother. And they exert their effect on the embryo regardless of the genotype of the father, because they were produced in the egg cell, in the cytoplasm of the egg cell that after fertilization becomes the zygote. So it is the mother that determines the initial stages of cell differentiation. Now, this polarity inside of the zygote, after subsequent divisions, sets up also a polarity in the embryo. So the polarity in the embryo means that not all the cells are going to have exactly the same contents. Now here's a second experiment done on, I think, the embryo of a, was it a sea star? No, I think it's a sea urchin, which is a relative of the sea star. Okay, so what did this experiment consist of? Well, we have a egg cell that is being fertilized by a sperm cell. This egg cell has an unequal distribution of cytoplasm on the inside, animal pole, vegetal pole. This cell, after fertilization, will divide, produce a two-cell stage. Then it will continue to divide, produce a four-cell stage, which continues to divide, and we get an 8-cell stage. Now at this point we have 8 cells, and we could ask ourselves, well, did the polarity of the cell already set up a polarity of this very early embryo? And here is what the experimenters did. They carefully separated the cells along this plane. That is, they separated the cells from the animal pole from the cells in the vegetal pole. Or in another experiment, they cut right through, maintaining the polarity of the embryo with the animal pole and the vegetal pole. And then they were looking for, well, what happens? And this is what they found in the results. When they separated the cells from the vegetal pole from those of the animal pole, we got either a cell that remained embryonic and that was characterized by this proliferation of cilia, so this cell didn't do anything further, or you got an abnormal larva. It looked like a larva, but it wasn't quite normal. On the other hand, when we maintained the polarity by cutting through the vertical plane, we did get normal larvae, although they were a little small. They were quite normal. So here we can see in this particular organism, the cell fate has already been determined by the polarity of the embryo, which is due to the polarity in the early zygote. Pretty cool, huh? All right. So, the internal environment of the cell of the original zygote is very important. But it also turns out that the external environment is very important. That is, by which types of cells a particular cell is surrounded with. And that will determine exactly what happens in terms of the development of this particular cell. So in this case, we're looking at a frog embryo. And in green here, we have the developing forebrain, and we have some surface tissue. Now, as the brain develops, the optical vesicle, which is this bulge right here, produces some molecules, messenger molecules, that will cause the surface tissue to become thicker and to develop what's called a lens placoed tissue. Now, the optic vesicle continues to develop. It forms a cup, and it continues to induce these cells to become the lens, so that eventually this tissue here will become the lens that's inside of the eye. The lens in turn induces these other cells on the surface tissue to become the cornea. Now, think about this. If these cells of the optic vesicle or the optic cup are important in inducing these cells to become the lens, what happens if I insert a barrier in between these cells? What do you think? What do you think happens? All right. Will the lens continue to form, or will lens formation stop? We can talk about this in class. This process of some cells determining the fate of its neighboring cells is called induction. Okay? All right. Now, I really don't want to talk in a lot of detail about this particular picture, and you can read up on this in the textbook, but it is worthwhile just to spend a little bit of time talking about this. The organism that you see up here is Caenorhabditis elegans. It's one of the model organisms used for all sorts of research, from the biomedical sciences to ecology and evolutionary studies. And what's so cool about this particular species is that all adults consist of just 959 cells. That's less than 1,000 cells. Can you imagine? Now, with an organism that is quite complex, it has a pharynx where it ingests food and digests food. It has a digestive tract. Here's the intestine. It has an ovary. Well, in this particular species, it has both male and female organs. We have a complex organism. Okay, so what is so cool about this species then is that with so few cells in the adult, we can actually map out exactly which cells will turn into some other type of cell. And this has been done, for example, with those cells that will turn out to form part of the vulva, which is the structure through which these worms lay their eggs. Now, these cells begin with an anchor cell. Let me get my pointer back. So here's the anchor cell. The anchor cell produces a primary inducer that will activate a gene that will cause these cells to become precursor cells for the vulva. Now, these cells in turn, these ones out here, will activate or upregulate gene 2, the cells, these here on the outside, but this cell won't because of its position of exactly where it is. And we can follow these cells through development. We can see that these here become secondary precursors, and these ones in the middle become the primary precursors. To make a long story short, we can actually follow what each cell becomes, and we can study exactly which inducer will produce these changes. Now here, these cells that never received the inducer from the anchor cell, well, they just become epidermis. All right, read up on this in the text. Now, the next very important class of molecules that is very, very important for differentiation of cells are called morphogens. Now, morphogens are signal molecules that provide positional information within the developing animal. Remember that first experiment that we talked about. The position of where we inserted those cells that normally would become skin cells. We changed their position. And now with a different position, they became something else. So these morphogens provide positional information. They tell the cell where in the body it is. Now, in order to classify as morphogen, a molecule has to directly affect target cells rather than produce a secondary signal. So the effect is directly on the target cell, and its effect is in proportion to its concentration. That is, a small concentration will lead to a certain change, a larger concentration to a different change. So the concentration is actually very important. It's not an all or nothing kind of signal. And this is actually a very important point because usually morphogens are produced by certain cells, and from these cells they diffuse down the body axis. Obviously, the concentration close to these cells that produce the morphogen are going to be higher, and then through diffusion, as we move away from these cells, the concentration will drop. Okay, here's an example of what this may look like in some organisms. And in this particular case, the example is from Drosophila melanogaster. Now, you should at this point know what Drosophila melanogaster is. It's the fruit fly, so it's an insect. Okay. Here we go. There are a couple of morphogens that are very important in setting up the polarity of the developing larva. And these morphogens are called bicoid and nano. Now it turns out that bicoid, when in high concentrations, results in the formation of the head and the thoracical segments. Now, an insect is a highly segmented organism. The nanomorphogen is responsible for producing the abdominal segments. Now together, these two morphogens set up the anterior posterior axis of the body. Let's look at this graph down here. On the y-axis we have concentration from low to high, and on our x-axis we have the position of a particular cell in the embryo. Here we have the anterior of the embryo, here we have the posterior of the embryo. We can see that the concentration of bicoid protein is very high at the beginning, that is at the anterior end, and then it drops, whereas the pattern for Nano's protein is reversed. It's very high in the rear, that is the posterior of the embryo, and then drops as we move towards the anterior end. All right. Obviously, different concentrations. These different concentrations are going to have a different effect on the cells. All right. Now, after this polarity of the developing larvae has been set up, other genes come into play, and we have two types of genes here: segmentation genes and homeotic genes. The segmentation genes are important for determining segments along the body of the developing fly. Gap genes organize broad areas along the anterior-posterior axis. Interestingly, if a mutation occurs in a gap gene, the resulting individual through development may actually miss several larval segments. Now, after the gap genes, the pair rule genes become important. They divide the embryo into units of 2 segments each. Okay, now mutations here will cause every other segment to miss in the embryo. It's no longer a large section, but a small section that's going to be missing. And then finally, we have segment polarity genes, which determine the boundaries and the anterior-posterior organization of the segments. So all of these genes act in concert. I'm going to show you on the next slide. After the segments have been already determined, then homeotic genes kick in, and they will define exactly what's going to happen to each segment. They're going to determine if a segment has a leg sticking out of it or an antenna. Now, interestingly, these homeotic genes are a large class of genes that we can also find in plants. It's these homeotic genes that include Hox genes, and these Hox genes are unique to animals. They control what happens to the segments, for example, in mammals. Now, usually Hox genes come in clusters of genes. And they always look, one of the genes looks like the next one, looks like the next one. They're all very similar, which is why we believe that they originally arose from gene duplication. Now what's interesting, as the anterior part of the animal develops and may differentiate its segments, it is the first genes in the sequence that is active. Then as we move back to segments following the head, now another gene turns on in sequence. And as we move from front to back of the animal on the chromosome in a linear direction, these Hox genes are turned on and turned off. Now, what happens if a Hox gene is turned on out of sequence? If it is turned on at the wrong time. Now, there are some really interesting mutations that arise from this wrong timing in turning on these genes. One of those is called the antennapedia, and the other one is called the bithorax. Okay, antennapedia. I actually mentioned this already in the last class. Antenna, well, that's the antenna, and pedia is foot. Okay, so we have, what do we have? We have, in fact, instead of having an antenna, we have a leg sticking out from exactly the same position. That is because the Hox gene for leg was turned on at the wrong time. It was turned on when the Hox gene determining antenna had to turn on. So just by changing the timing of turning on and off the Hox genes, we will create these types of mutations. The bithorax mutation, I don't have a picture here for you, is a mutation in which an additional thoracic segment produces wings. Now, usually in a fly, you have 2 wings and 2 modified little wings that help more for balance and flight. But in the bithorax mutation, now we have 4 functional wings on the fruit fly. And that is because the Hox gene for wing formation turned on at the wrong time on one of the segments that normally does not have any wings. Alright, so now let's look at this. Over here on this side, you can see how one of the morphogens determines what happens to the next type of gene, what happens to the next. So we start out with our maternal effect genes, our bicoid and nanos. Bicoid high concentration here, nanos here. That sets up the polarity of the developing embryo. Now it turns out that bicoid has an effect on another gene. Effectively, it turns on the hunchback gene and turns off the Krüppel gene. That's a German word, Krüppel gene. Now, obviously, it will really turn hunchback on here and Krüppel off where the concentration is very high. But then as the concentration becomes lower, the effect on these 2 genes, hunchback and Krüppel, which are gap genes, is going to be much different. Nanos, on the other hand, which is at a high concentration back here, is going to inhibit the translation of hunchback. Now this is actually pretty cool because in a way here bicoid will affect transcription factors, hunchback, so it will inhibit transcription, whereas nanos inhibits translation. That means that the hunchback mRNA is already present here, but nanos inhibits its translation. So even though there may be hunchback turned on here by bicoid, nanos may actually inhibit its translation so that hunchback never becomes something else. Now, these gap genes, hunchback and Krüppel, will turn on or turn off pair rule genes. And the pair rule genes are transcription factors. These transcription factors are regulated by the gap genes, and the gap genes already define or produce these different areas. Every of these areas is going to have a different mix of concentration of gap genes, which then will lead to differences in the pair rule genes. And eventually, these pair rule genes, which produce transcription factors, will regulate the segment polarity genes. So we can see there's a cascade of events that all begins with this polarity here. If we were to eliminate polarity in the early developing embryo, then we would affect the entire rest of the development. And then, last not least, the homeotic genes will determine what happens exactly to each one of these segments that has been produced by all of this cascade of events. Wow. Okay, now a note about Hox genes. Hox genes, like all homeotic genes really, are so-called homeobox containing genes. The homeobox is actually a sequence of DNA of about 180 base pairs. Now, when transcribed, when translated, these 180 base pairs of DNA are going to produce 60 or so amino acids. And the sequence of amino acids is a DNA binding motif. That is, this particular sequence has a very high affinity for DNA on those segments of the gene in the control region that will determine whether or not a particular gene is transcribed. Now, this DNA binding motif, well, it's actually part of transcription factors. Remember that many transcription factors need to bind to DNA in order to activate transcription. So in this particular case, since the Hox genes contain the homeobox which produces a DNA binding motif, well, it already tells us that these particular Hox genes, they encode transcription factors. Now, just to give you an example, the bicoid homeodomain, that is the domain that recognizes the DNA, it actually recognizes this specific sequence in the control region. Alright, so these transcription factors tell us exactly when and where to start transcription. So up to this point we have seen how maternal effect genes set up a polarity inside of the zygote and the developing embryo. And well, we've seen that these maternal effect genes, they actually produce oftentimes morphogens that provide a sense of location for each cell within the developing embryo. But development is actually a little more than just simple differentiation, and it turns out that programmed cell death or apoptosis is actually also a very important component of development. So any gene that will trigger apoptosis in certain cells, well, is going to be very important as well. Now, one example we can find in the developing human embryo, for example. 41 days after fertilization of the zygote, the embryo has, in between its digits, still skin tissue expressed. So there's actually tissue in here, and it reminds us perhaps of an amphibian ancestor that at some point we may have had. But obviously we are no longer amphibians. We use our hands not so much for swimming as we use them for manipulation. So this tissue between the fingers is actually quite it's actually quite a hindrance for what we use our hands for. So this particular tissue then has to undergo through apoptosis. It has to kill itself off so that we have separate fingers as we have them now on our hands. Now, it's a mechanism to undo previous evolutionary adaptations. So the controlled death of certain cells that then can no longer develop or become something, they just disappear. So just as a little note that apoptosis, programmed cell death, is an important part of development.